Hello everyone and a warm welcome to my presentation. I'm Negar and I'm about to take you on an adventure into the fascinating topic of decoding the mystery of memory, with a special focus on an incredible insight from Aplysia's nervous system. Who here has ever experienced that frustrating tip of the tongue moment, where a memory teases you, but it stays just out of reach? Or on a brighter note, who's been amazed at how a song from years ago can instantly transport you back in time? These moments highlight the profound role of memory. It's not just a function of the brain, but the essence of an organism's survival toolkit. It helps us to shape our ability to adapt to an ever changing world. Today, I'm here to take you on a journey into the fascinating topic of learning and memory by first introducing our humble yet extraordinary model organism, Aplysia, and exploring its nervous system advantage for neuroscience studies. Then we'll explore the phenomenon of sensitization, a form of learning and memory observed in Aplysia. Finally, we'll unravel the mystery of memory storage using Aplysia as our key to understanding. Now that we set the stage for the fascinating world of memory and learning, let's zoom in on the Sherlock Holmes of neuroscience, the remarkable Aplysia. Aplysia, often referred to as a sea hare, is a gastropod mollusk, a marine snail that lacks an external shell. The name sea hare comes from its distinctive large anterior tentacles, resembling ears and leading to a strange comparison with a hare. Fascinatingly, Aplysia considered a genus of medium to large-sized sea slug, boasting an impressive diversity of approximately 37 recognized species, ranging in size from just a few centimeters to an astonishing 60 to 70 centimeters. These sea slugs are a true marvel of marine world. Hold on to your hat, folks, because Aplysia isn't just your average marine creature. They are hermaphrodite and they have both male and female reproductive organ, and they don't settle for ordinary. They form a chain of up to 20 individuals. The seer at the front act as a female, and the one at the back act as a male, and those in the middle are both male and female, inseminate the partner in front of them while receiving sperm from the one behind them. Scientists are all over the moon about Aplysia, and it's not because of their impressive dance move in the mating scene. No, it's all about their brain power. The steer nervous system is made of a small number of very large neurons, which make it very easy to study them. Their nervous system organized into 9 ganglia, hosting around 20,000 neurons. It might sound like a lot, but when we compare it to the grand scale of vertebrate, especially human who have an impressive 86 billion neurons on average, it's a relatively small number. Also, seer nervous system is easily accessible and neurons have a distinctive personality, where most of them assigned with a unique name like R2 or R14, as you can see in the photo. Aplysia neurons, especially the R2 neuron, are not just large, they're colossal. Imagine fitting an entire Drosophila brain, or a dozen C. elegans worm into a single neuron. These giant neurons are not only the largest among somatic cells in the animal kingdom, but also display a vivid natural coloration, from orange and yellow to milk, reddish, or even greenish. But why are they so big? It turns out their neurons carry extensive amount of genetic material compared to other cells. Natural tissue taken from Aplysia has a really cool ability to stay alive and send signals for a long time. This adaptability comes from the fact that Aplysia are living in a tide pool, shallow water, rocky and sandy substrate, where the temperature, saltiness and tide changes a lot. Because Aplysia doesn't have a fixed body temperature and its soft body can quickly adapt to changes, its nervous system becomes super strong. When we take out the nervous system from Aplysia, it can stay active at room temperature without needing anesthesia. It has gradual respiratory rate that allows it for extensive study without significant change in their function. A form of memory that has been well studied in a sea hare is called sensitization of gill and siphon withdrawal reflex. But what is sensitization? Picture Aplysia and a sensitive siphon and gill. When a tactile stimulus is applied to the siphon, the Aplysia defensively withdraws its gill, an adaptive mechanism crucial for fending off predator and ensuring survival. Now imagine a scenario. The sea slug faces an intense jolt, a tail shock. It's like a wake-up call for the aplysia that heightens sensitivity. The gill and siphon become supercharged and responsive to the tiniest ripple in their surrounding. This heightened sensitivity is more than a mere reaction. It represents the learning experience called sensitization. Through this encounter, the aplysia form a memory that boosts its responsiveness in the future. With repeated exposure to the noxious or strong stimuli, the aplysia can develop a long-term sensitization, or you can even say a long-term memory because it results in the sea slug exhibiting heightened sensitivity to its surrounding for days or months. You can also see the difference between normal and sensitized animal in their reaction to a light touch to their siphon. The normal animal barely show a reaction, while you can see how much more powerful the withdrawal is for the sensitized one. Researchers found that long-term sensitization is due to synaptic plasticity. Synapse is a connection between nervous system cells called neuron, and plasticity is like a strengthening this connection between neurons. When the sea slug experiences something intense repeatedly, like a strong stimulus, the connection between the neuron gets stronger. It's like building a stronger bridge between the cells in the nervous system to hold on into important memories. Mechanism of memory storage is a complex and ongoing area of research. The exact nature of engram, the physical substrate that containing our memory, remain uncertain. Although there is no definite answer yet, the common idea suggests that long-term memory are likely housed in the synapses. However, some researchers believe they are stored in your non-coding RNA. RNA is best known for helping your cell create protein, but non-coding RNA isn't involved in making protein. There are different types of non-coding RNA, and we're still figuring out what they can do. In a recent research, there is some evidence that some of their job can be in storing memory. In that study, scientists give shock to the tail of the sea slug and produce the long-term sensitization. As you can see, the control group didn't perform a significant siphon withdrawal reflex, while the sensitized group had an average of 60 seconds of siphon withdrawal reflex, showing that they're sensitized. Then scientists took sample of RNA from the sea slug nervous system of both control and trained group and injected it into the circulatory system of another group of sea slug. The sea slug with the injection of RNA from sensitized animal also performed long-term sensitization, although they were not treated with a tail shock before. This showed that sea slug had been basically injected with a memory of the first group. After follow-up experiment, researchers believe that non-coding RNA trigger DNA methylation. This involves attaching methyl group to a specific DNA region, typically leading to a suppression of certain protein production. This alteration may in turn affect sea slug neuron. The research findings indicate that inhibiting DNA methylation during sensitization training prevent memory formation. Also, inhibiting DNA methylation after training, even when the sensitization memory has already formed, lead to the erasure of that memory. There is still a lot to figure out, like how non-coding RNA causes methylation, and how methylation changes sea slug neuron and ultimately memory. As we wrap our journey, let's reflect on the key takeaway. The unique trait of aplysia with its small number of large neurons have provided us with valuable insight into the working of memory, learning, and neurobiology. The seer has proven to be a fascinating model organism, offering a glimpse into the complexity of nervous system. The exploration of sensitization in Aplysia has unveiled a captivating form of learning that not only impacts the seer's behavior, but also provides a window into the broader mechanism of memory and learning. While the exact location of memory engram remains elusive, our journey has highlighted the prevailing theory of synaptic plasticity and non-coding RNA. these theories present the intriguing possibility for understanding how memory are stored and retained within Aplysia's nervous system. In conclusion, Aplysia's nervous system has served as a captivating subject, challenging us to explore the boundary for our knowledge. Thank you for joining me, and I hope you enjoyed.